So now I want to talk about muscle structure and contraction. Here's a muscle showing a bundle of muscle fibers here. Muscle fiber is a muscle cell. So each muscle cell is a muscle fiber. We call them muscle fibers because they're long and thin. And in a vertebrate, they're very long. They're among the largest cells in your body. They can be many centimeters long. They're multinucleate. They actually form by fusion of individual cells. And that's because of our large size. That's unusual for animals. Within each muscle cell are myofibrils, long skinny strands that have sarcomeres within them. So there are a row of sarcomeres that are in a series. So here I'm showing one sarcomere. That one sarcomere is composed of actually quite a few different types of protein, but the critical ones that we're going to talk about for function are the myosin, a bundle of myosin, and here are the myosin heads. So the little gray loops there are myosin heads. The green is actin. So actin filaments. Actin is actually a round globular protein that is assembled into these long filaments, and nebulin is a protein that actually is twisted along with it, and it arranges and keeps the length of the actin filaments the right length. So we won't talk about it much more, but without the nebulin, the actin filaments wouldn't be the right length, wouldn't hold together properly. Titin is another protein, and it arranges and holds the myosin bundles in place. Okay, critical for function is also in blue. I'm showing tropomyosin. That would be all along here. Tropomyosin covers the binding sites of actin, and the result of it is that as long as tropomyosin is there in skeletal muscle, contraction can't happen. Now that's going to be different when we talk about smooth muscle, so don't get confused by that. In red is troponin. Troponin is a calcium binding protein, same family as calmodulin. And it, once calcium binds, that changes its conformation, and that causes tropomyosin to pull away from the from the actin binding sites. Now I'm going to run through the cycle of myosin contraction. We start in the cocked position, in which an ATP has been split into ADP and phosphate. Here are the binding sites in black. I'm showing 4 actin molecules in a row within a series. Of course, there'd be many more, and it's actually a double twisted strand of actins. Tropomyosin is out of the way here. So with tropomyosin out of the way, what happens is that the myosin head is now free to bind. Here's our same 4 actins. I added an extra one at the end. And with myosin, with tropomyosin out of the way, it can attach. When the attachment happens, that destabilizes the position for phosphate. So phosphate actually pops free in this new conformation. And once phosphate is free, the myosin head is no longer stable in the cocked position. So with phosphate gone, that myosin head flips to a new conformation, and that moves the actin relative to the myosin. So here we are in the bent conformation called the rigor conformation, and actin has moved, and now in this new conformation, in the rigor conformation, ADP pops free. It no longer sticks. But myosin is stuck in the rigor position, bound to actin, until it can bind a new ATP. So a new ATP, once one comes in and binds to that binding site, now with that ATP there, myosin is no longer stable when it's connected to actin, and so it detaches from actin in this new conformation. Once it's detached, that ATP, the shape of the binding pocket for ATP changes, and that pushes free, it pops free the phosphate into a new binding pocket, into a nearby binding pocket. So now you have an ADP and a phosphate. With this phosphate now, -2 in a new position, that causes the myosin head to recock. It pushes the myosin back into that recocked position, and we're back where we started. ADP, phosphate head, but notice the position of our actins. Before we bound to position 2, but in the next cycle of contraction, and many cycles happen with each muscle twitch, each firing of a muscle, the next cycle of contraction we're going to bind to actin 3 or 4, and we're going to continue to move actin to the left and myosin to the right. So if I just showed you a schematic with some way too large actins, here's some myosins, so a bundle of myosins, they bend and pull, and so after a few cycles what we'd have is a shortened sarcomere. And when you shorten all the sarcomeres, you shorten the muscle. So how is this regulated? In skeletal muscle, each cell is regulated by synapses with neurons. So here's a synapse with acetylcholine, a neurotransmitter. And those skeletal muscle cells have receptors for acetylcholine, and the acetylcholine receptor is a ion channel. It's an ionotropic receptor for sodium. And so when acetylcholine binds, sodium enters, moving down its charge gradient and into an area with lower concentration. Of course, that sodium, as it disperses and other positive ions dispersing away, that opens voltage-gated sodium channels. Sodium comes in, and just like we've discussed many times, we get a new action potential that spreads. I've shown here these little indentations. They're actually little tubes that go into the cell, and they function to bring the action potential deep into the cell. These are big cells, remember, and if we didn't have these tubules, these T or transverse tubules, we wouldn't get the action potential down into near the deep sarcomeres. That's important because voltage change opens voltage-gated calcium channels. It's at sarcoplasmic reticulum. So these blue circles represent a network of what's called sarcoplasmic reticulum. It's basically a type of endoplasmic reticulum that's specialized to store calcium. Calcium pumps fill them with calcium. Keep that in mind. This is slow moving calcium in. Calcium channels allow calcium out. That's much faster, so contraction starts quickly, but is much slower to stop. And then there are also calcium-activated calcium channels, so calcium-regulated calcium channels. So a little bit of calcium coming in from these voltage-gated channels allows much more calcium out. Okay, so that brings calcium out. The action potential spreads, we get these voltage-gated channels open and calcium being released, and slowly after the AP, calcium is returned to the sarcoplasmic reticulum. The calcium-activated calcium channels are inactivated after being open for a while. Okay, so we've got a sarcomere here near some sarcoplasmic reticulum. It's near enough to a T-tubule, transverse tubule, to have brought in the action potential to open nearby sarcoplasmic reticulum. Tropomyosin in red here, covering the actin binding site. And on that tropomyosin are troponins, calcium binding protein. Calcium has been released by the action potential, calcium binds, and that pulls the tropomyosin away from those actin binding sites. So here's some myosin head next to an actin again. There's the binding sites. Tropomyosin's been pulled away. There's, as long as calcium is bound there, so calcium bound to troponin, we're going to get the actin binding sites exposed. So contraction happens, and in fact there will be attachment of myosin and continued cycling and continued movement of actin relative to myosin until the calcium is removed.